So here's something worth thinking about as you settle in tonight. Right now, at this very moment, there are electrical signals racing through your body at speeds that would put most internet connections to shame. Some of them are traveling at over 270 miles per hour. Others are crawling along at barely a mile per hour. And the difference between those fast ones and those slow ones, the reason some signals sprint and others take their sweet time, that's one of the most elegant engineering stories in all of biology. But we'll get to that. Let's start somewhere quieter. Let's start with what people used to think was happening inside the body. Because honestly, the history of how we figured out the nervous system is one of the strangest, most winding roads in all of science. And it begins, like so many things do, with the ancient Greeks getting something almost right and something spectacularly wrong at the same time. Around 400 BC, there was a physician on the Greek island of Kos named Hippocrates. You've probably heard his name. He's the one behind the Hippocratic Oath that doctors still reference today. And Hippocrates had this radical idea, radical for his time anyway, that the brain was the organ responsible for sensation and intelligence. He wrote about it pretty directly. He said the brain is the interpreter of consciousness, which, sounds obvious to us now, but at the time, this was genuinely controversial. Because the dominant view, the one that would actually win out for centuries after Hippocrates, was that the heart was the seat of the mind. Aristotle, who came along a generation or so later, and who was far more influential in the long run, looked at the brain and basically dismissed it. He thought it was a cooling organ, like a radiator. He noticed that the brain didn't seem to respond when you touched it directly, and he noticed that it was full of fluid, and he figured its job was to cool the blood that was heated by the heart. The heart, Aristotle argued, was where thought and sensation actually happened. And if you think about it, you can see why someone might believe that. When you're scared, you feel it in your chest. When you're in love, your heart pounds. When you're startled, it's your heart that leaps. The brain just sits there, quietly, doing nothing you can feel. So right from the beginning, there's this fundamental confusion. Where does sensation live? Where does the mind live? And that confusion isn't trivial. It shaped medical practice for over 1,000 years. If you think the heart is the seat of consciousness, you're not going to spend much time investigating the brain, are you? And that's exactly what happened. But there were people who pushed back. One of the most important was a man named Galen, a Greek physician who lived in the 2nd century AD and spent much of his career in Rome patching up gladiators. And I love this detail because it matters for the story. Galen's experience with gladiators gave him something almost no other physician of his era had, which was extensive first-hand knowledge of what happens when specific parts of the body get damaged. He saw what happened when a sword cut through a particular nerve. He saw which injuries caused paralysis and which caused loss of sensation. He was, in a very real sense, learning neuroanatomy from combat injuries. And what Galen concluded, based on years of this grim but invaluable observation, was that Hippocrates had been right. The brain was the command center, not the heart. Galen went further, though. He developed a theory about how the brain actually communicated with the rest of the body. And this is where things get interesting and also, honestly, a little bit wrong in a very understandable way. Galen believed in what he called animal spirits, spiritus animalis in Latin. The idea was that the brain produced a kind of subtle fluid, almost like a vapor or a very fine liquid, and this fluid flowed through the nerves, which Galen correctly identified as the physical connections between the brain and the body. He thought the nerves were hollow tubes, like tiny pipes, and that the animal spirits flowed through them the way water flows through an aqueduct. When you wanted to move your arm, your brain would pump animal spirits down through the nerve to the muscles of your arm, and the muscles would inflate, like little balloons, and that's what caused movement. Now, here's the thing. This is wrong. Nerves are not hollow tubes. Muscles don't inflate. There are no animal spirits. But, and this is what I find so fascinating, the basic framework isn't that far off. There is a signal that travels from the brain, down through the nerves, to the muscles. There is a physical pathway, and the signal does cause the muscles to contract. Galen had the architecture roughly right. He just had the mechanism completely wrong. And that distinction between getting the architecture right and the mechanism wrong, that's going to come up again and again in this story. Because the nervous system is so complex that for most of human history, people could see what it did without having any real idea how it did it. And Galen's theory stuck around for a remarkably long time. We're talking well over 1,000 years. Through the Middle Ages, through the Renaissance, the idea of animal spirits flowing through hollow nerves was basically the accepted model. Even Leonardo da Vinci, who dissected human bodies and drew the most extraordinary anatomical illustrations anyone had ever seen, even Leonardo basically accepted the animal spirits framework. He made wax casts of the ventricles of the brain, those fluid-filled chambers deep inside, because he believed that's where the animal spirits were produced. And his drawings of the ventricles were actually more accurate than anything that had come before. But the underlying theory was still Galen's. So let's sit with that for a moment. For roughly 1,500 years, from Galen in the 2nd century to the 1600s, the best model anyone had for how your nervous system worked was essentially a plumbing system. Brain makes fluid, fluid flows through pipes, muscles inflate. It's a hydraulic model, and you can see why it was so persistent, right? It's intuitive. It maps onto things people already understood. Romans built aqueducts. They understood hydraulics. The metaphor made sense. But then something happened in the 1600s that started to crack the whole thing open, and it came from an unexpected direction. It came from frogs. In 1664, a Dutch scientist named Jan Swammerdam did something beautifully simple. He took a frog, he dissected out a leg muscle with the nerve still attached, and he put the muscle in a glass container. Then he irritated the nerve, just pinched it basically, and watched the muscle contract. So far, nothing surprising. Galen would have predicted that. But here's what Swammerdam did that was clever. He sealed the glass container so that a tiny drop of water sat in a narrow tube at the top, like a very sensitive volume indicator. If the muscle was inflating, if it was filling up with animal spirits the way Galen said, then the total volume inside the container should increase, and that drop of water should rise. But it didn't. The muscle contracted, it got shorter and thicker, but the drop of water didn't move. The total volume stayed the same. The muscle wasn't inflating. Nothing was flowing into it. Whatever was causing the contraction, it wasn't a fluid filling the muscle up like a balloon. This was a quiet little experiment. Swammerdam didn't even publish it during his lifetime. His notes were found after he died. But it was one of those moments where a very simple observation starts to unravel a very old idea. If animal spirits weren't inflating the muscles, then what was happening? What was the signal that traveled down the nerve? And this is where we need to talk about electricity. Because the real answer, the one that would take another century and a half to fully emerge, is that the nervous system is electrical, not hydraulic, not mechanical, electrical. And the story of how that was discovered involves one of the most famous scientific rivalries in history: a dead frog, a thunderstorm, and two Italian scientists who genuinely could not stand each other. The year is 1780, and we're in Bologna, Italy. Luigi Galvani is a physician and anatomist at the University of Bologna, and he's been doing experiments on frogs. There are a lot of frogs in this story, by the way. Frogs were basically the lab rats of early neuroscience because their legs would twitch very visibly when you stimulated the nerves, and they were easy to get. So Galvani has a dissected frog laid out on a table, and the story goes, and the exact details are debated, but the core of it seems to be true, that a metal scalpel touched the frog's sciatic nerve while, nearby, a static electricity machine was generating sparks, and the frog's leg kicked, just twitched, right there on the table, a dead frog's leg, moving as if it were alive. Galvani was fascinated. He started experimenting systematically. He hung frog legs from iron railings with brass hooks during thunderstorms and watched them twitch when lightning flashed nearby. He tried different metals, different conditions, and he came to a conclusion that was, in its own way, as revolutionary as anything in the history of biology. He believed he had discovered what he called animal electricity, an electrical force that was inherent to living tissue itself. The animal spirits of Galen, Galvani was saying, weren't a fluid at all. They were electrical. The body generated its own electricity, and that's what flowed through the nerves. Now, this is actually pretty close to the truth. But Galvani had a problem, and his problem's name was Alessandro Volta. Volta was a physicist at the University of Pavia, about 120 miles away, and he thought Galvani's interpretation was wrong. Volta argued that the electricity wasn't coming from the frog at all. It was coming from the junction between the two different metals, the brass hooks and the iron railing. The frog's leg was just a very sensitive detector. It was the metals generating the electricity, not the animal tissue. And so began one of the great scientific arguments of the 18th century. Galvani and Volta went back and forth, publishing papers, conducting experiments, each trying to prove the other wrong. Volta eventually built his famous voltaic pile, essentially the first battery, to prove that you could generate electricity from metals alone, no frog required. And Volta was right about that. The battery worked. You didn't need living tissue to make electricity. But here's the kicker. Galvani was also right. Living tissue does generate its own electricity. Both of them were correct about different things, and both of them were wrong about what the other one was claiming. Galvani actually proved his own point beautifully in a later experiment that doesn't get enough credit. He took a frog's leg, and, without using any metal at all, he draped the sciatic nerve directly over the cut end of the leg muscle, and the leg twitched. No metal, no external electricity, just nerve-touching muscle, and the leg moved. That was animal electricity, pure and simple. The tissue itself was generating an electrical signal. But Galvani died in 1798, impoverished and largely discredited, while Volta went on to fame and glory. Napoleon himself was so impressed by Volta's battery that he made him a count. The unit of electrical potential, the volt, is named after Volta. And Galvani? Well, we use his name too. When you galvanize someone into action, that's him. When you galvanize steel, that's him. But for a long time, the scientific establishment sided with Volta, and the idea that living tissue had its own electricity was pushed to the margins. It would take decades before the scientific world came back around to what Galvani had been trying to say. And the person who really brought it back, who proved beyond any doubt that nerves carry electrical signals, was a German physiologist named Émile Dubois-Reymond, working in Berlin in the 1840s. Dubois-Reymond was meticulous, almost obsessively careful, and he built incredibly sensitive instruments, galvanometers, that could detect the tiny electrical currents produced by living nerve and muscle tissue. And in 1843, he demonstrated something remarkable. He showed that when a nerve was stimulated, there was a measurable change in its electrical state, a wave of electrical activity that traveled along the nerve. He called it the negative variation, which isn't the catchiest name, but what he was describing was nothing less than the nerve impulse itself. The actual signal that your brain sends down your nerves to move your muscles, to feel the world, to do everything your nervous system does. Dubois-Reymond was the first person to measure it directly. And that changes everything. Because once you know the signal is electrical, you can start asking the real questions. How fast does it travel? What generates it? How does it get from one nerve cell to the next? And perhaps, most importantly, how does a nerve cell, which is not a copper wire, which is not a metal conductor, which is a soft, wet, biological thing, how does it conduct electricity at all? Because here's the thing that would puzzle scientists for another 100 years. Nerve signals are electrical, yes, but they don't work anything like the electricity in your house. Not even close. The mechanism is completely different, and figuring out how it actually works required a giant squid, a pair of brilliant English scientists, and some of the most delicate experiments ever attempted on living tissue. So let's talk about that. The problem, if you really sit with it, is genuinely strange. You've got a nerve fiber, right? It's basically a long, thin tube filled with salty water, surrounded by more salty water. That's it. That's your wiring. And if you know anything about electricity, you know that salt water is a terrible conductor compared to copper. Signals in a copper wire travel at nearly the speed of light, something like 200 million meters per second. Signals in your nerves? Depending on the nerve, maybe 1 meter per second on the slow end, maybe 120 meters per second on the fast end. That's not even in the same universe. So whatever nerves are doing, it's not just passively conducting electricity the way a wire does. Something else is going on, something active. And for a long time, nobody could figure out what that something was. Now, to understand how scientists eventually cracked this, you need to know a little bit about the cells themselves. By the late 1800s, people were starting to get a much clearer picture of what the nervous system actually looked like under a microscope, and this is where another one of those great rivalries comes in, one that shaped neuroscience for decades. On one side, you had Camillo Golgi, an Italian physician working in Pavia. On the other side, Santiago Ramón y Cajal, a Spanish anatomist working mostly in Barcelona and Madrid. And these two men fundamentally disagreed about the basic architecture of the nervous system. Golgi had developed an incredible staining technique. He discovered it almost by accident in the 1870s, working in a hospital kitchen of all places, experimenting with silver chromate. And what this stain did was remarkable. For reasons nobody fully understood, it would randomly pick out maybe 1 or 2% of the neurons in a tissue sample and dye them completely black, leaving everything else transparent, which meant that for the first time, you could see the full shape of individual nerve cells, these extraordinary branching structures with long projections reaching out in every direction. Before Golgi's stain, looking at nervous tissue under a microscope was like looking at a plate of spaghetti. Everything was tangled together, impossible to tease apart. His technique changed that overnight. But here's the irony. Golgi himself looked at those beautiful images and concluded that the nervous system was one continuous network, a single connected web of fibers, like a fishing net, where everything was physically fused together. He called it the reticular theory, and it made a certain kind of intuitive sense. If signals need to get from point A to point B quickly, why wouldn't the system be wired as one continuous mesh? Kajal, using Golgi's own staining method, and this is what makes the story so delicious, looked at the same kind of images and came to the exact opposite conclusion. He argued that each nerve cell was a separate, individual unit, that they came close to each other, incredibly close, but they didn't actually touch. They were discrete entities, like individual links in a chain rather than threads in a net. This became known as the neuron doctrine. And Kajal was right. We know that now. But at the time, it was a genuine scientific war. These two men argued for years, published competing papers, gave competing lectures, each absolutely convinced the other was wrong. And then, in 1906, they were jointly awarded the Nobel Prize in Physiology or Medicine. Both of them, together, for their work on the structure of the nervous system. You can imagine how that banquet went. By most accounts, neither man was particularly gracious about sharing the honor. Golgi actually used his Nobel lecture to attack Kajal's theory. Which is, that takes a certain kind of stubbornness, right? You're at the Nobel ceremony, and you're still arguing. But the reason this matters for our story about how nerve signals work is that Kajal's insight, that neurons are separate cells, raised an enormous question. If nerve cells don't physically touch each other, how does a signal get from one to the next? There must be some kind of gap, some junction point, and that gap, which the English physiologist Charles Sherrington would name the synapse around 1897, became one of the great mysteries of the early 20th century. We'll come back to the synapse later, because it's a whole world of its own. But for now, let's stay with the individual nerve cell and the question of how it carries a signal along its own length. Because even within a single neuron, the electrical behavior was baffling. By the early 1900s, scientists knew several things that didn't quite add up. They knew the signal was electrical. They knew it traveled at a fixed speed that was way too slow to be simple conduction. And they knew something else that was really puzzling. The signal didn't get weaker as it traveled. Think about that. If you send an electrical pulse down a cable underwater, it fades. The signal degrades. Resistance eats it up. But a nerve impulse travels from your spinal cord all the way down to your big toe. That's roughly a meter. And it arrives just as strong as when it started. That's not how passive electrical conduction works. The nerve must be doing something to regenerate the signal along the way, boosting it as it goes, like a series of amplifiers laid end to end. And this is where the giant squid enters the picture. Sometimes the history of science turns on the strangest details, and this is one of my favorites. In the 1930s, a young English zoologist named J.Z. Young was studying squid at a marine biological station in Naples, and he noticed something that other people had seen before, but hadn't fully appreciated. Squid have these enormous nerve fibers running along their bodies, part of their jet propulsion escape system. When a squid needs to flee from a predator, it contracts its mantle and shoots water out in a jet. And it needs to do this incredibly fast, which means the nerve signals controlling that contraction need to travel very quickly. And one of the ways nature solved that problem was by making the nerve fiber really, really big. We're talking about an axon. That's the long projection of a nerve cell that carries the signal. That's up to a millimeter in diameter, maybe even a little more. Now, a millimeter doesn't sound like much, but most of your nerve axons are maybe 1 or 2 micrometers across, 1,000th of a millimeter. The squid giant axon is roughly 1,000 times wider. You can see it with the naked eye. You can pick it up with tweezers. And J.Z. Young realized that this was a gift to science, because all the delicate electrical measurements that people wanted to make on nerve fibers, all the things that were essentially impossible on the tiny, fragile axons of mammals, you could actually do on this thing. You could stick an electrode inside it. You could measure what was happening across its membrane in real time. He published his findings about the squid giant axon in 1936, and he essentially told the world, here is your experimental preparation. Here is how you can finally figure out how the nerve impulse works. And the people who took him up on that invitation were Alan Hodgkin and Andrew Huxley, 2 young physiologists at Cambridge. Now, Hodgkin had actually started working on nerve impulses before the war, and he'd already made a crucial observation. In 1939, working with the squid giant axon at the Marine Biological Association laboratory in Plymouth, he and Andrew Huxley managed to do something remarkable. They inserted a fine glass electrode directly inside the axon and measured the electrical voltage across its membrane. And what they found was surprising. The inside of the nerve cell, when it was just sitting there at rest, was electrically negative compared to the outside, about -70 millivolts. There was a voltage difference across the membrane even when the nerve wasn't doing anything. That resting potential, as it's called, was like a tiny battery, just sitting there, waiting. But then, the war intervened. Hodgkin went off to work on radar. Huxley worked on gunnery, and the squid would have to wait. It wasn't until 1945 that they could get back to Plymouth, back to the squid, back to the question that had been nagging at them for 6 years. And when they did, they brought better equipment, sharper questions, and a determination to solve this thing once and for all. Here's what they needed to explain. When a nerve impulse passes a given point on the axon, the voltage across the membrane doesn't just decrease a little, it flips. It goes from that resting -70 millivolts all the way up to about +40 millivolts, and then it snaps back down again. The whole event takes about a millisecond. And this spike, this brief, dramatic reversal of voltage, is the action potential. That's the nerve impulse. That's the signal. Every thought you've ever had, every sensation you've ever felt, every movement you've ever made, it's all built from these tiny voltage spikes racing along your nerve fibers. But what causes the spike? What makes the voltage flip like that? This is where Hodgkin and Huxley's work becomes truly extraordinary, because they figured out the mechanism in almost complete detail, using equipment that by modern standards was laughably primitive. No computers, no digital oscilloscopes, just analog electronics, patience, and a technique called the voltage clamp, which Hodgkin and Huxley refined to an art form. The idea behind the voltage clamp is clever. Instead of just watching what the voltage does during an impulse, you force the membrane to sit at a specific voltage and then measure what currents flow across it. It's like pinning the voltage in place and asking, okay, at this particular voltage, what are the ions doing? What's flowing in, what's flowing out, and how fast? And ions are the key to everything. Because remember, we said the nerve is basically a tube of salty water inside more salty water. But the salt inside isn't the same as the salt outside. The fluid outside the nerve cell is rich in sodium ions, positively charged sodium atoms. The fluid inside is rich in potassium ions, also positively charged, but a different element. And the cell membrane, that thin, fatty layer that wraps around the nerve cell, is normally almost impermeable to both. It holds them apart. Sodium outside, potassium inside, and never the twain shall meet, at least not until the nerve decides to fire. And what Hodgkin and Huxley discovered through years of painstaking experiments between 1947 and 1952 was that the membrane contains tiny protein channels, molecular gates, that can open and close in response to voltage changes. When the membrane voltage reaches a certain threshold, sodium channels snap open, and sodium ions come flooding into the cell. Positive charges rushing inward. That's what drives the voltage from negative to positive. That's the rising phase of the action potential. And then, a fraction of a millisecond later, those sodium channels slam shut, and a different set of channels, potassium channels, open up, letting potassium ions rush out of the cell. Positive charges leaving, and that's what brings the voltage back down again. The beauty of it is that it's self-propagating. When one little patch of membrane fires, when sodium rushes in and the voltage spikes, that voltage change spreads to the neighboring patch of membrane, which triggers those sodium channels to open too, which triggers the next patch and the next, like a row of dominoes falling, or better yet, like a fuse burning along its length. Each section ignites the next. The signal doesn't fade because it's being actively regenerated at every point along the way. The energy isn't coming from the original stimulus, it's coming from the ion gradients themselves, from that careful separation of sodium and potassium that the cell maintains at enormous metabolic cost, pumping ions back and forth 24 hours a day, 7 days a week, even while you sleep. Especially while you sleep, actually, because your brain doesn't shut off when you close your eyes. Right now, as you're lying there listening to this, billions of action potentials are firing across your nervous system, maintaining your heartbeat, regulating your breathing, consolidating your memories from today. The pumps never stop. Hodgkin and Huxley published their model in 1952 in a series of 5 papers in the Journal of Physiology, and the final paper included a set of mathematical equations that described the whole process, how the sodium and potassium channels open and close, how the currents flow, how the action potential propagates. They solved those equations by hand, mostly, with Huxley grinding through the calculations on a hand-cranked mechanical calculator. It took him weeks to compute what a modern laptop could do in seconds. And when they plotted the results, the predicted shape of the action potential matched the real thing almost perfectly. It was one of those rare moments in science where a theoretical prediction lines up so cleanly with experimental data that you know you've found something true. They won the Nobel Prize for it in 1963, shared with John Eccles, who we'll get to later because his story ties into what happens at the synapse, that gap between neurons that Kajal and Sherrington had identified decades earlier. And understanding what happens at that gap is really the next piece of the puzzle, because so far we've been talking about how a signal travels along a single neuron from one end to the other, like a flame racing down a fuse. But your nervous system isn't one neuron. It's billions of them, and they have to talk to each other. The signal has to jump from one cell to the next across that tiny gap, the synapse, and how it does, that turns out to be one of the most elegant and surprising mechanisms in all of biology. So let's talk about John Eccles. He's the third name on that 1963 Nobel Prize, and his story is fascinating partly because he spent years passionately defending an idea that turned out to be wrong, and then, to his enormous credit, he did the experiments that proved himself wrong, and he accepted it. That takes a kind of intellectual courage that's genuinely rare. Eccles was Australian, born in Melbourne in 1903, and he went to Oxford on a Rhodes Scholarship in the 1920s, where he studied under Charles Sherrington, the same Sherrington who coined the word synapse back around 1897. So Eccles was trained by the man who first named the problem, and he inherited Sherrington's deep interest in how signals cross that gap. The question was straightforward. When an action potential reaches the end of one neuron, how does it trigger a response in the next neuron? What's the messenger? And there were two camps. One camp said it was electrical. The signal is electrical inside the neuron, so why wouldn't it just jump electrically across the gap? Maybe the current from the arriving action potential is enough to directly trigger the next cell. Simple, clean, fast. Eccles was firmly in this camp. He believed synaptic transmission was fundamentally an electrical process. The other camp said it was chemical. They believed the arriving signal caused the first neuron to release some kind of chemical substance into the gap, and that chemical then drifted across and triggered the next neuron. This idea had been floating around since the early 1900s, and it had a champion, a German pharmacologist named Otto Lowy. Here's the thing about Lowy's story. It's one of those tales that sounds almost too good to be true, but it's well documented. In 1921, Lowy was living in Graz, Austria, and he later said that the idea for his crucial experiment came to him in a dream. He woke up in the middle of the night, scribbled some notes, went back to sleep, and in the morning, he couldn't read his own handwriting, couldn't remember the dream either. But the next night, the dream came back. This time he got up, went straight to his laboratory. It was Easter Saturday, reportedly, and performed the experiment right then and there, in the middle of the night. What he did was beautifully simple. He took 2 frog hearts. He stimulated the vagus nerve on the first heart, which slowed it down. That was already well known, that stimulating the vagus nerve slows the heart. Then he took the fluid that was bathing that first heart and transferred it to the second heart. And the second heart slowed down too, even though nobody had touched its vagus nerve, even though there was no electrical connection between the 2 hearts. Something in the fluid, some chemical released by the nerve stimulation of the first heart, was carrying the message. Lowy called this substance Vagusstoff, which just means Vegas stuff in German. It was later identified as acetylcholine, a molecule that turns out to be one of the most important neurotransmitters in your entire body. Lowy won the Nobel Prize for this in 1936, shared with Henry Dale, who had done crucial work identifying acetylcholine and showing that it was released at various nerve endings. So by the 1930s, there was solid evidence that at least some synapses used chemical transmission. But Eccles wasn't convinced that this applied everywhere, especially not in the fast synapses of the brain and spinal cord. And honestly, his skepticism wasn't unreasonable. Lowy's experiment involved the autonomic nervous system, the part that controls your heart rate, your digestion, those slow, involuntary processes. Chemical transmission seemed plausible there because those processes are relatively slow. But the fast reflexes in your spinal cord, the rapid signaling in your brain, Eccles argued those were too fast for chemicals. By the time a chemical diffused across the gap, got recognized by the other side, triggered a response, it would take too long. It had to be electrical. This debate went on for years. Eccles was vocal, articulate, and deeply committed to the electrical hypothesis. He and his colleagues published papers arguing their case. It was a genuine scientific controversy, the kind where smart people looked at the same evidence and drew different conclusions. And then, in the early 1950s, right around the same time Hodgkin and Huxley were publishing their action potential model, Eccles did something remarkable. He developed techniques for recording from individual neurons inside the spinal cord of a cat using incredibly fine glass microelectrodes. These electrodes were so thin you could insert them directly into a single motor neuron and measure what was happening inside the cell when a signal arrived at the synapse. Now, if transmission were purely electrical, you'd expect the response in the receiving neuron to look a certain way. It should be almost instantaneous. It should follow the timing of the incoming signal very precisely, and it should have certain electrical characteristics. But that's not what Eccles found. What he saw was a small delay, about half a millisecond, between the arrival of the signal and the response in the next cell. And the response had properties that were consistent with a chemical process, not a direct electrical one. The shape of the signal was different. The timing was different. The way it could be modified was different. Eccles looked at his own data and realized he'd been wrong. Chemical transmission wasn't just for slow autonomic processes. It was the primary mechanism at fast synapses too. And he published these findings honestly and clearly, essentially dismantling his own previous position. He later said that Karl Popper's philosophy of science had influenced him. The idea that you should try to falsify your own hypotheses, not just confirm them. Whether that's the whole story or a bit of retrospective framing, the fact remains that Eccles did the hard thing. He followed the evidence. So here's how chemical synaptic transmission actually works, and it's worth walking through step by step because it's genuinely beautiful engineering, or, well, biological engineering, shaped by hundreds of millions of years of evolution, but engineering nonetheless. An action potential comes racing down a neuron and reaches the very end, the axon terminal. This terminal is sitting right next to the next neuron, separated by that tiny synaptic gap, which is only about 20 nanometers wide. 20 nanometers. That's about 1/5000th the width of a human hair. Inside the axon terminal, there are these little spherical packages called synaptic vesicles, and each one is loaded with molecules of a neurotransmitter, acetylcholine, or dopamine, or serotonin, or glutamate, or any of dozens of different chemical messengers, depending on what kind of neuron we're talking about. When the action potential arrives, it does something clever. It opens voltage-gated calcium channels in the terminal membrane. Not sodium channels this time, calcium channels. Calcium ions rush in from outside, and that influx of calcium is the trigger. It causes the synaptic vesicles to move to the membrane, fuse with it, and dump their contents into the synaptic gap. This process is called exocytosis, and it happens remarkably fast, within a fraction of a millisecond. Now the neurotransmitter molecules are floating in that tiny gap, and they drift across. It doesn't take long. The gap is so narrow, and they bump into receptor proteins on the surface of the next neuron. These receptors are incredibly specific. They're shaped to recognize one particular neurotransmitter, like a lock that fits only one key. When the neurotransmitter binds to the receptor changes shape, and this opens an ion channel in the membrane of the receiving neuron. Ions flow through, and the electrical state of the receiving neuron changes. And here's where it gets really interesting, because the change can go in two directions. If the synapse is excitatory, the neurotransmitter opens channels that let positive ions in, typically sodium. And this pushes the receiving neuron closer to firing its own action potential. But if the synapse is inhibitory, the neurotransmitter opens channels that let negative ions in, typically chloride, or let positive ions out, and this pushes the receiving neuron further away from firing. It makes it harder to trigger. Think about what that means. Every neuron in your brain is receiving inputs from thousands of other neurons simultaneously, some saying fire and some saying don't fire, and the neuron has to integrate all of those signals, add them up, and decide whether the total is enough to reach threshold and trigger its own action potential. It's like a vote. Thousands of tiny votes, some yes and some no, happening every millisecond. And the neuron fires only if the yeses outweigh the nos by enough. This is what Sherrington had intuited decades earlier when he talked about excitation and inhibition in the spinal cord, watching how reflexes worked in animals. He could see that some inputs facilitated a response and others suppressed it. He just didn't know the mechanism. Now, with Eccles's work and the work of many others through the 1950s and 1960s, the mechanism was becoming clear. And the cleanup is just as important as the signal itself. After the neurotransmitter has done its job, it has to be removed from the synapse quickly, or it would just keep stimulating the receiving neuron over and over. There are several ways this happens. Enzymes in the synaptic gap can break the neurotransmitter down. Acetylcholinesterase, for instance, chops acetylcholine into pieces so fast that each molecule of the enzyme can process thousands of acetylcholine molecules per second. Or the neurotransmitter can be pumped back into the original neuron and recycled, which is called reuptake. That word, reuptake, might sound familiar because it's the target of some of the most widely prescribed drugs in the world. Selective serotonin reuptake inhibitors, SSRIs, drugs like fluoxetine and sertraline, work by blocking the reuptake of serotonin so it stays in the synapse longer and has a greater effect. Millions of people take these drugs for depression and anxiety, and the mechanism they target is exactly this synaptic cleanup process that Eccles and his contemporaries were just beginning to understand in the 1950s. So now we've got the basic picture. Signals travel along neurons as electrical impulses, action potentials, and they cross the gaps between neurons as chemical signals, neurotransmitters. Electrical, then chemical, then electrical again. It's a hybrid system, and each step has its own logic, its own speed, its own vulnerabilities. But there's a deeper question lurking here, and it's one that fascinated researchers throughout the mid-20th century. We've been talking about individual neurons and individual synapses, but how do you get from that to something like a thought, or a memory, or the ability to learn a new skill? How does the system change over time? Because clearly it does change. You weren't born knowing how to ride a bicycle or speak a language. Something in your neural circuitry is being modified by experience. And figuring out what that something is, how the connections between neurons can be strengthened or weakened by use. That story starts with a Canadian psychologist named Donald Hebb, who in 1949 published a book called The Organization of Behavior. And in that book, he proposed an idea so simple and so powerful that it fundamentally reshaped how we think about learning and memory at the level of the brain. Hebb's idea, boiled down to its essence, goes something like this. When one neuron repeatedly helps fire another neuron, the connection between them gets stronger. That's it. That's the core insight. Neurons that fire together wire together. That's the catchy version you might have heard, though Hebb himself never actually used that phrase. It was coined later as a kind of mnemonic summary. What Hebb actually wrote was more careful and more nuanced, but the spirit is the same. If neuron A consistently participates in making neuron B fire, then something changes at the synapse between them. some growth process or metabolic change that makes A more effective at triggering B in the future. Now think about what that means. It means the brain isn't a fixed circuit. It's not like a computer motherboard where the connections are soldered in place at the factory. The connections between your neurons are plastic. They can be strengthened, weakened, even created or destroyed, depending on how they're used. And this plasticity, this capacity for change, is what allows you to learn. Every time you practice a piano scale, every time you study a new word, every time you navigate a familiar route home, the relevant neural pathways are being subtly reshaped. The synapses that matter for that task are getting a little bit stronger, a little bit more efficient, a little bit more likely to fire in the right pattern the next time. Hebb proposed this in 1949, and here's the thing. He had almost no direct experimental evidence for it. He was a theorist, working from behavioral observations and logical inference. He couldn't actually measure synaptic strength changing in a living brain. The technology simply didn't exist yet. He was making a prediction, and it was a bold one. A lot of his contemporaries were skeptical. The book didn't exactly set the world on fire when it first came out. It sold modestly. Some reviewers were enthusiastic, but others thought it was too speculative, too hand-wavy, not grounded enough in hard data. But Hebb's background is worth knowing because it explains why he was thinking about these questions in the first place. He'd studied under Carl Lashley at Harvard, and Lashley had spent decades trying to find the physical location of memory in the brain. Lashley's approach was brutally direct. He would train rats to run mazes, then surgically remove different parts of their cerebral cortex and see which lesion destroyed the memory. And the maddening result, over and over again, was that no single lesion could completely erase a learned behavior. You could cut out a chunk of cortex, and the rat would still remember, maybe a little worse, but still functional. Remove more cortex and performance degraded further, but it degraded gradually, proportionally to how much you removed, not in a sudden all-or-nothing way. Lashley called this finding mass action, the idea that memory seemed to be distributed across the cortex rather than stored in one specific spot. This drove Lashley a little bit crazy, honestly. He spent his career looking for the engram, the physical trace of a memory, and at the end of it, he famously quipped that sometimes he felt the only conclusion was that learning was simply not possible. He was joking, obviously, but the frustration was real. He couldn't find the filing cabinet. There was no single drawer labeled maze memory that you could pull out and destroy. Hebb took this failure and turned it into an insight. Maybe the reason you can't find memory in one place is that memory isn't in one place. Maybe memory is in the pattern of connections between neurons, in the network, not in any individual cell. A memory, in Hebb's framework, is a cell assembly, a group of neurons that have been strengthened into a functional unit through repeated co-activation. When you remember something, you're reactivating that assembly. The memory lives in the relationships between the neurons, in the synaptic weights, not in the neurons themselves. That's why Lashley couldn't destroy a memory with a single cut, because the assembly was distributed, and even a damaged network could partially reconstruct the pattern. This was a genuinely revolutionary way of thinking about the brain, and it took decades for the experimental evidence to catch up. But when it did, it came in a spectacular way. The year was 1973, and the place was Oslo, Norway. Two researchers, Terje Lomo and Tim Bliss, published a paper that would become one of the most cited in all of neuroscience. What they found was this. If you stimulated a neural pathway in a rabbit's hippocampus with a brief burst of high-frequency electrical pulses, the synapses along that pathway became stronger, and they stayed stronger. Not for seconds, not for minutes, for hours. In later experiments, for days, even weeks. They called this phenomenon long-term potentiation, or LTP, and it was, in essence, the physiological confirmation of Hebb's theoretical prediction from 24 years earlier. Let me walk you through what's actually happening, because the mechanism is beautiful. In the hippocampus, which, by the way, is a seahorse-shaped structure deep in your temporal lobe that's absolutely critical for forming new memories, there are synapses that use glutamate as their neurotransmitter. On the receiving side of these synapses, there are two types of receptors that matter here. One is called the AMPA receptor, and the other is called the NMDA receptor. Under normal conditions, when a signal comes through at a regular low-frequency rate, only the AMPA receptors respond. They open up, let sodium ions flow in, and generate a normal postsynaptic response. The NMDA receptors are there too, but they're blocked. There's a magnesium ion sitting in the channel like a cork in a bottle, and at normal resting conditions, it won't budge. But here's what's clever. If the postsynaptic neuron is already partially depolarized, already somewhat excited, because it's receiving a lot of input at the same time, then the electrical change across its membrane is enough to knock that magnesium ion out of the NMDA receptor channel. And when the NMDA receptor opens, it lets in not just sodium, but also calcium. And calcium, inside a neuron, is a signal. It's a trigger. It sets off a cascade of molecular events. Enzymes are activated, proteins are phosphorylated, gene expression is altered, and the end result is that more AMPA receptors get inserted into the membrane of that synapse. More receptors means the synapse is now more sensitive to glutamate. The same amount of neurotransmitter produces a bigger response. The connection has been strengthened. And notice the logic of it. The NMDA receptor only opens when two things happen simultaneously. The presynaptic neuron releases glutamate, and the postsynaptic neuron is already active. It's a coincidence detector. It's detecting exactly the situation Hebb described. Two neurons firing together. That's why people sometimes call the NMDA receptor a Hebbian synapse. It's the molecular implementation of the rule Hebb proposed on purely theoretical grounds in 1949. Now, LTP is just one side of the coin. If synapses could only get stronger, the whole system would eventually saturate. Every connection cranked up to maximum, no contrast, no signal, just noise. So there has to be a way to weaken connections too. And there is. It's called long-term depression, or LTD, and it's essentially the reverse process. Low-frequency stimulation, the kind that doesn't strongly activate the postsynaptic neuron, leads to a modest, slow trickle of calcium through NMDA receptors, enough to activate a different set of enzymes that actually remove AMPA receptors from the synapse. The connection gets weaker, so the brain has a bidirectional tuning mechanism. Synapses can be dialed up or dialed down depending on the pattern of activity. Strengthen what's useful, weaken what's not. It's elegant, and this connects directly to something you experience every single day. When you learn someone's name and remember it a week later, LTP-like processes in your hippocampus helped encode that memory. When you forget the name of someone you met once at a party 3 years ago, that's partly because those synaptic changes weren't reinforced and gradually faded. The system is always editing itself, always updating. But here's where the story gets richer, and honestly, a little heartbreaking. The person who taught us the most about the hippocampus and memory wasn't a scientist. He was a patient. His name was Henry Molaison, though for decades he was known in the medical literature only by his initials, HM, to protect his privacy. His full identity wasn't revealed until after his death in 2008. Henry suffered from severe epilepsy. By his late 20s, in the early 1950s, his seizures were so frequent and so debilitating that he couldn't hold a job or live a normal life. The seizures were originating from his medial temporal lobes, and in 1953, a neurosurgeon named William Beecher Scoville performed an experimental operation. He removed large portions of Henry's medial temporal lobes on both sides, including most of both hippocampi. The surgery worked, in the sense that it dramatically reduced Henry's seizures, but it also produced a devastating side effect that no one had fully anticipated. Henry could no longer form new long-term memories. He could remember his childhood. He could remember events from before the surgery, at least the older ones. He could hold a conversation, and his intelligence was intact. His IQ actually went up slightly after the surgery, probably because he wasn't being constantly disrupted by seizures. But the moment a conversation ended and you left the room, he would forget you. You could walk back in 5 minutes later, and he would greet you as a stranger. He lived in a permanent present tense, every day a fresh page, every face a new face. The neuropsychologist Brenda Milner studied Henry for decades, beginning in 1957, and her work with him produced some of the most important findings in the history of memory research. Because here's what was surprising. Henry wasn't completely unable to learn. Milner gave him a task called mirror tracing, where you have to trace the outline of a star while looking only at your hand's reflection in a mirror. It's awkward and difficult at first, and Henry got better at it, day after day, with practice. His hand became steadier, his tracings more accurate, but every single day he had no memory of ever having done the task before. He would sit down and say he'd never seen this apparatus in his life, and then perform it with the skill of someone who'd been practicing for days, because he had been. What this told Milner, and eventually the whole field, was that there isn't just one kind of memory. There are at least two fundamentally different systems. The kind that Henry lost, the ability to remember events, facts, names, what you had for breakfast, that's called declarative memory or explicit memory, and it depends critically on the hippocampus. The kind he retained, the ability to learn motor skills, to improve with practice, to acquire habits, that's called procedural memory or implicit memory, and it depends on different brain structures, particularly the basal ganglia and the cerebellum. Two different systems, two different anatomies, two different kinds of knowing. And this distinction matters in ways that go far beyond the laboratory. Think about someone with Alzheimer's disease, where the hippocampus is one of the first structures to deteriorate. They may forget your name, forget that you visited yesterday, forget where they are, but they might still be able to play a song on the piano that they learned 50 years ago. The procedural system is still intact, even as the declarative system crumbles. That's Henry Molaison's legacy, in a way. His tragedy illuminated a fundamental architecture of the mind. But memory isn't the only thing the nervous system has to manage. While researchers in the 1950s and 60s were mapping out the hippocampus and the synapse, a parallel line of investigation was unfolding. One focused not on how we remember, but on how we sense the world in the first place. How does the brain take the raw electrical signals pouring in from your eyes, your ears, your skin, and turn them into a coherent experience? How do you see a face, hear a melody, feel the warmth of a cup of tea? And the answers that began emerging from this work were, if anything, even more surprising than what Milner found in Henry's damaged memory. Because it turned out that the brain doesn't passively receive information from the senses. It actively constructs what you experience. It builds the world you think you're seeing, hearing, touching, assembles it piece by piece from incomplete, noisy, sometimes contradictory data. And the story of how we figured that out starts, fittingly, with a cat, a darkened laboratory, and two researchers who were trying to do something else entirely. David Hubel and Torsten Wiesel were working at Johns Hopkins in the late 1950s, and they were interested in the visual cortex, the part of the brain at the back of your head that processes what your eyes take in. Their setup was straightforward in concept, though painstaking in practice. They would anesthetize a cat, prop its eyes open, and insert a tiny electrode into individual neurons in the visual cortex. Then they'd show the cat different things, spots of light, dark shapes on bright backgrounds, and listen for which neurons fired in response. They were using an audio monitor, so when a neuron fired, they'd hear a little click or pop through a speaker. Silence meant the neuron wasn't interested. A burst of clicks meant they'd found something that neuron cared about. And for a while, they found almost nothing. They'd project spots of light onto a screen in front of the cat's eyes, and the neurons in the visual cortex just sat there, mostly quiet. Occasionally one would fire a little, but nothing dramatic, nothing consistent. They spent hours, days, trying different spots, different sizes, different positions. It was frustrating work, the kind of research where you start wondering if your equipment is broken, if your hypothesis is wrong, if you're wasting your time. And then, according to the story Hubel told many times afterward, something accidental happened. They were sliding a glass slide into their projector, a slide with a dark spot on it that they'd been using as a stimulus, and as the edge of the slide passed through the projector beam, it cast a faint line of light across the screen. And suddenly, the neuron they were recording from went absolutely wild. A burst of rapid firing, loud and unmistakable through the speaker. They moved the slide again. Same response. They tried orienting the line differently, tilted it, rotated it, and the neuron went quiet. Put it back at the original angle, and there it was again, firing like crazy. Here's what they had stumbled onto. That neuron didn't care about spots of light. It cared about a line at a specific orientation. A bar of light tilted at, say, 45 degrees would make it fire intensely. Tilt that bar to 50 degrees, and the response dropped off. Tilt it to 90 degrees, and the neuron barely responded at all. It was tuned. It was selective. It had a preference. And this was just the beginning. Over the next several years, through the early 1960s, Hubel and Wiesel mapped out an astonishing hierarchy in the visual cortex. They found what they called simple cells, which responded to edges and bars at specific orientations in specific locations of the visual field. Then they found complex cells, which also cared about orientation, but were less fussy about exact position. A bar at 45 degrees would excite them whether it was a little to the left or a little to the right. And then there were hypercomplex cells, which responded to oriented lines of specific lengths, or to corners, or to edges that stopped at a certain point. Think about what this means. Your visual cortex isn't just receiving a picture from your eyes like a camera sensor receiving an image. It's disassembling the visual scene into features, edges, orientations, corners, movement directions, and processing those features through successive layers of increasingly sophisticated neurons. The raw input from your retina, which is really just a pattern of light and dark hitting millions of photoreceptor cells, gets broken down and rebuilt. First into oriented edges, then into combinations of edges, then into shapes, then further along in the cortex, into objects, faces, places. It's like the brain is running an assembly line, but in reverse. Instead of starting with a finished product and breaking it down, it starts with the rawest possible material, photons hitting your retina, and builds up layer by layer toward the rich visual experience you're having right now. Except you're lying in the dark. So maybe the experience you're having right now is more about the inside of your eyelids. But you get the idea. Hubel and Wiesel won the Nobel Prize in 1981 for this work, and they deserved every bit of it, because what they revealed wasn't just about vision, it was a general principle. The brain processes sensory information hierarchically. Simple features first, then combinations, then more abstract representations. And this principle turns out to apply across all the senses. In the auditory cortex, you find neurons tuned to specific frequencies of sound, and then higher-order neurons that respond to combinations of frequencies, to frequency sweeps, to the kinds of complex patterns that make up speech or music. In the somatosensory cortex, the strip of brain that processes touch, there are neurons tuned to specific types of pressure, specific locations on the skin, specific textures. And here's the thing that really gets interesting. The way these processing areas are organized in the brain isn't random. There are maps. Your somatosensory cortex contains a map of your entire body surface, but it's a distorted map. The parts of your body that are most sensitive, your fingertips, your lips, your tongue, take up a disproportionately huge amount of cortical real estate compared to, say, your back or your elbow. If you drew a human figure where each body part was sized according to how much brain is devoted to sensing it, you'd get this bizarre, grotesque little creature with enormous hands, a massive tongue, and a tiny torso. That figure actually has a name, the cortical homunculus. And if you've ever seen a picture of it, you know it's wonderfully weird looking. It was mapped out by a neurosurgeon named Wilder Penfield in the 1930s and 40s, working in Montreal, actually not too far from where Brenda Milner would later study Henry Molaison. Penfield's method was extraordinary and, honestly, a little unsettling to think about. He performed brain surgery on epileptic patients who were awake. The brain itself has no pain receptors. You can touch the surface of the brain and the person feels nothing there. So once the skull was opened under local anesthesia, Penfield could electrically stimulate different points on the cortex and simply ask the patient what they experienced. Touch this spot, and the patient says, "I feel a tingling in my right thumb." Touch that spot, something on my lower lip. Touch another spot, and the patient might hear a sound or see a flash of light, or, and this is the part that really captivated people, suddenly experience a vivid memory. One patient reported hearing a specific song playing, as if a record had been put on. Another recalled being in a specific place from childhood. Penfield called these experiential responses, and they suggested that the brain stores experiences in a way that can be replayed, almost like a recording. Now, modern neuroscience has complicated Penfield's interpretation quite a bit. Those vivid memories triggered by electrical stimulation probably weren't perfect recordings being played back. They were more likely reconstructions, the brain's associative machinery being jolted into producing something that felt like a memory, but might have been partly fabricated. And this connects to something really important about how the nervous system works, something that applies not just to memory, but to perception itself. Your brain is not a passive recorder. It's not a camera. It's not a microphone. It's a prediction machine. And this idea that the brain is fundamentally in the business of making predictions about what's going to happen next, and then checking those predictions against incoming sensory data, has become one of the most influential frameworks in modern neuroscience. It goes by several names: predictive processing, predictive coding, the Bayesian brain hypothesis. The details vary depending on who you talk to, but the core insight is the same. Here's how to think about it. At every level of the sensory processing hierarchy that Hubel and Wiesel mapped out, the brain isn't just passively waiting for information to arrive from below. It's actively sending predictions downward. Higher level areas are constantly telling lower level areas, "Here's what I think you're about to see. Here's what I think you're about to hear." And the lower level areas compare those predictions against the actual incoming data. If the prediction matches, not much happens. The signal is suppressed because there's nothing new to report. But if the prediction is wrong, if something unexpected shows up, then an error signal gets sent upward, and the higher levels update their model. Think about what that means for your everyday experience. Most of the time, your brain is basically running a simulation of the world and checking it against reality. When you walk through your house in the middle of the night, you don't need to carefully examine every wall and door frame. Your brain already has a model of your house, and it's predicting what's there. You navigate almost on autopilot. But if someone moved a piece of furniture, you'd notice immediately. You might even bump into it, because the prediction didn't match, and that mismatch generated a big error signal that grabbed your attention. This is why magic tricks work. The magician is deliberately manipulating your predictions. They set up a pattern. Look, the ball goes into this hand, see? And your brain starts predicting that the ball will continue to be in that hand. When the prediction is violated, you experience surprise, wonder, the feeling of impossibility. But nothing impossible happened. Your prediction engine just got outmaneuvered. And this predictive framework explains something that would otherwise be deeply puzzling about the nervous system: the sheer volume of feedback connections. If the brain were a simple bottom-up processor, data comes in from the senses, gets processed through successive stages, and produces a perception at the top. Then you'd expect most of the connections to run upward from lower areas to higher areas. But that's not what the anatomy shows. In the visual cortex, for instance, there are actually more connections running from higher areas back down to lower areas than there are running upward. For decades, nobody could really explain why. The predictive processing framework gives those feedback connections a job. They're carrying predictions downward, while the feed-forward connections carry prediction errors upward. Now, this idea didn't come out of nowhere. It has roots going back to the 19th century, to Hermann von Helmholtz, the same Helmholtz who measured the speed of nerve signals, the one we talked about earlier. Helmholtz proposed that perception is a process of unconscious inference. He argued that what you see isn't simply what's on your retina. It's a conclusion that your brain draws, an inference, based on the retinal image, plus everything your brain already knows about the world. You're not seeing raw data, you're seeing an interpretation. And Helmholtz was right, in ways that are now supported by enormous amounts of evidence. Consider optical illusions. The reason illusions work is precisely because your brain is making inferences, and sometimes those inferences are wrong. The famous Müller-Lyer illusion, those two lines with the arrowheads pointing in or out, where one line looks longer than the other, even though they're the same length. That happens because your brain is interpreting the arrowhead shapes as depth cues, the kind you'd see at the corners of rooms, and adjusting its estimate of line length accordingly. The brain isn't making a mistake, exactly. It's applying a rule that works brilliantly in the three-dimensional world and just happens to get fooled by a flat drawing. So here you are, lying in the dark, and your nervous system is still doing all of this. Even with your eyes closed, your brain is running predictions about the sounds in the room, about the position of your body, about what's going to happen next. It's constructing a model of the world and updating it moment by moment. And the raw material for all of this construction, all of this prediction, all of this inference is still just electrochemical signals running along neurons and jumping across synapses, the same basic machinery that Hodgkin and Huxley described with their equations, the same sodium and potassium channels, the same neurotransmitter vesicles fusing with membranes. But there's another dimension of the nervous system we haven't really explored yet, one that's running quietly in the background all the time, even now, keeping you alive without you having to think about it. Your heart is beating. Your lungs are breathing. Your gut is digesting whatever you ate for dinner. And none of that requires your conscious attention, because there's an entire division of the nervous system dedicated to handling it, a division with its own fascinating history, its own set of discoveries, and its own surprising complexities. It's called the autonomic nervous system, and the story of how we came to understand it begins with a question that seems almost too simple. Why does your heart speed up when you're scared? Think about it. Nobody tells their heart to beat faster. You don't consciously decide to sweat when you're nervous, or to get goosebumps when you're cold, or to dilate your pupils when you walk into a dark room. These things just happen, and for most of human history, that was deeply mysterious. This idea that parts of your body seem to have a mind of their own, operating on some hidden set of instructions that you couldn't access or override. The ancient physicians had noticed it, of course. Galen talked about different kinds of pneuma, animal spirits flowing through the nerves to control voluntary movement, and a separate natural spirit governing the organs. It was a rough distinction, but it captured something real. There genuinely are two different systems at work. The nerves that let you wiggle your fingers are fundamentally different from the nerves that speed up your heart, and untangling that difference took centuries. The term autonomic itself comes from the early 20th century. It was coined by John Newport Langley, a Cambridge physiologist who spent decades mapping out the nerves that control the organs. Langley was meticulous, almost obsessively so. Starting in the 1890s, he used nicotine, actual nicotine, applied directly to nerve fibers to trace the pathways of what he called the autonomic nervous system. The word autonomic means self-governing, and that's exactly the idea. This is the part of your nervous system that governs itself, that runs the show without waiting for your conscious input. But Langley's real contribution was organizing the system into two opposing branches, and this is where it gets really elegant. He called them the sympathetic and the parasympathetic nervous systems, and the way they work together is one of the most beautiful pieces of biological engineering you'll ever encounter. Here's the simplest way to think about it. The sympathetic nervous system is your accelerator. The parasympathetic nervous system is your brake. They're both always active, both always exerting some influence, and the balance between them determines what your body is doing at any given moment. Right now, as you're lying there relaxing, your parasympathetic system is dominant. It's slowing your heart rate, promoting digestion, conserving energy. It's sometimes called the rest and digest system, which is a nice shorthand. Your body is in maintenance mode, repairing, processing, recovering. But if something startled you right now, a loud crash, an unexpected noise, your sympathetic system would take over in an instant. Heart rate up, breathing faster, blood diverted from your digestive organs to your muscles, pupils dilated, sweat glands activated, glucose released into your bloodstream for quick energy. This is the famous fight-or-flight response, and it's orchestrated by a cascade of signals running through sympathetic nerve fibers and amplified by hormones, especially adrenaline, or epinephrine if you prefer the more technical name, pouring out of your adrenal glands, which sit right on top of your kidneys. Now, the person who really nailed down the fight-or-flight concept was Walter Bradford Cannon, a Harvard physiologist working in the early 1900s. Cannon was a fascinating character. He was one of the first scientists to use X-rays to study digestion. He fed barium-laced food to cats and watched it move through their digestive systems in real time, which was absolutely cutting-edge stuff in 1897. But what he noticed during those experiments changed the direction of his career. When the cats became frightened or agitated, their digestion just stopped. The stomach contractions ceased. The whole digestive process ground to a halt. And Cannon realized he was watching the sympathetic nervous system override the parasympathetic system in real time. He spent the next several decades working out the details, and in 1915, he published his landmark work describing the fight-or-flight response as a unified physiological pattern. What Cannon understood was that all these individual changes, the racing heart, the shallow breathing, the cold sweat, the dry mouth, weren't separate reactions. They were a coordinated program, a single integrated response designed by evolution to prepare an animal for immediate physical action. Every change makes sense when you think about it from a survival perspective. Blood leaving your gut and flooding your muscles? You don't need to digest lunch if a predator is chasing you. Pupils dilating? You need to see as much as possible. Glucose flooding your bloodstream? Your muscles need fuel right now. And here's what's really clever about the engineering of this system. The sympathetic nervous system doesn't just use nerve signals. It also hijacks the endocrine system, the hormone system, to amplify and sustain its effects. The sympathetic nerves directly innervate the adrenal medulla, the inner part of those adrenal glands sitting on your kidneys, and when they fire, the adrenal medulla dumps adrenaline straight into your bloodstream. That adrenaline reaches every cell in your body within seconds, reinforcing and extending the same effects that the sympathetic nerves initiated locally. It's like the nervous system fires the opening shot, and then the hormones come in as sustained artillery. The result is a response that's both fast, because nerve signals travel in milliseconds, and long-lasting, because hormones circulate for minutes. Cannon also gave us another concept that's become absolutely foundational, not just in physiology, but in engineering and systems theory broadly. He called it homeostasis, the idea that your body is constantly working to maintain stable internal conditions, stable temperature, stable blood sugar, stable pH, stable fluid balance, and that the autonomic nervous system is the primary mechanism for doing it. Homeostasis isn't about keeping things perfectly constant. It's about keeping things within a livable range, using feedback loops that detect deviations and trigger corrections. Your body temperature drifts up slightly, and your autonomic nervous system triggers sweating and vasodilation to cool you down. It drifts down, and you get vasoconstriction and shivering. It's a thermostat, essentially, a biological thermostat running on nerve impulses and chemical signals. And this idea of feedback control, it shows up everywhere in engineering. Norbert Wiener, the mathematician who founded cybernetics in the 1940s, explicitly drew on Cannon's concept of homeostasis when developing his theories about self-regulating systems. The thermostat in your house, the cruise control in your car, the autopilot in an airplane, they're all doing what your autonomic nervous system does, measuring a variable, comparing it to a set point, and making adjustments. Your body was doing feedback control long before engineers invented the concept. But let's come back to the anatomy for a moment, because the physical layout of the autonomic nervous system is genuinely interesting, and it tells you something about how evolution solves problems. The sympathetic nerves emerge from the middle portion of your spinal cord, the thoracic and lumbar regions, roughly from your chest down to your lower back. They form a chain of ganglia, little clusters of nerve cell bodies that run like a string of beads along both sides of your spine. This is called the sympathetic chain, and it's a real physical structure you can see in a dissection, running from the base of your skull to your tailbone. From these ganglia, nerve fibers fan out to reach every organ, every blood vessel, every sweat gland in your body. The parasympathetic system, by contrast, takes a completely different route. Its fibers emerge from the brainstem, up at the top, and from the sacral region of the spinal cord, way down at the bottom. It's sometimes called the craniosacral system for this reason, and the most important parasympathetic nerve is one you might have heard of, the vagus nerve. The name comes from the Latin word for wandering, and it's perfect because this nerve wanders all over the place. It leaves the brainstem and travels down through the neck, into the chest, past the heart and lungs, and all the way down into the abdomen, branching along the way to innervate the heart, the lungs, the esophagus, the stomach, the intestines, the liver, the pancreas. It's the longest cranial nerve in the body, and it carries about 75% of all parasympathetic fibers. When your heart slows down as you relax, that's the vagus nerve applying the brake. When your stomach gurgles after a meal, that's the vagus nerve promoting digestion. There's a wonderful story about the vagus nerve that connects back to something we talked about earlier. Otto Loewy's dream experiment in 1921, the one where he stimulated a frog's vagus nerve and collected the fluid that slowed the heart, proving chemical neurotransmission. That fluid contained acetylcholine, the parasympathetic neurotransmitter. So Loewy's famous experiment wasn't just about proving that synapses use chemicals. It was specifically an experiment on the autonomic nervous system. The vagus nerve was his tool, and the heart was his readout. The autonomic system and the story of neurotransmission are woven together from the very beginning. Now here's something that might surprise you. For a long time, people thought of the autonomic nervous system as strictly a two-part system, sympathetic and parasympathetic, accelerator and brake, and that was it. But there's a third player that scientists increasingly recognize as its own semi-independent division, and it's been hiding in plain sight this whole time. It's called the enteric nervous system, and it lives in the walls of your gut. Your gastrointestinal tract, from your esophagus to your rectum, is lined with somewhere between 200 million and 600 million neurons. Let that number sink in for a second. That's more neurons than in your entire spinal cord. These neurons form two intricate networks, called the myenteric plexus and the submucosal plexus, woven into the walls of your digestive tract, and they can coordinate the complex muscular contractions of digestion entirely on their own, without any input from the brain or spinal cord. If you were to sever every nerve connecting the gut to the central nervous system, your intestines would keep right on working. They'd still push food along, still secrete digestive enzymes, still regulate blood flow to the intestinal lining. The enteric nervous system is so self-sufficient that it's sometimes called the second brain. And while that's a bit of an oversimplification, it captures something real about the degree of autonomy this system has. The person who first appreciated the complexity of the enteric nervous system was another one of those meticulous 19th century anatomists, Leopold Auerbach, a German neuropathologist who in 1862 described the nerve plexus in the muscular wall of the gut that still bears his name, the Auerbach's plexus. But it took over a century for scientists to really grasp how sophisticated this system is. In the 1990s and 2000s, researchers discovered that the enteric nervous system uses more than 30 different neurotransmitters, many of them identical to the ones used in the brain, including serotonin. And here's a detail that genuinely surprises most people. About 95% of the serotonin in your body is found not in your brain, but in your gut. 95%. That's the same serotonin that SSR is target for depression. The implications of that are still being worked out, and the relationship between gut chemistry and mood and mental health is one of the most active areas of research in neuroscience right now. But the basic point is that your gut has its own vast neural network, its own chemical signaling system, and a degree of independence that nobody suspected until relatively recently. So the autonomic nervous system turns out to be more complex, more layered, and more interesting than the simple accelerator and brake model suggests. You've got the sympathetic system revving things up, the parasympathetic system calming things down, the enteric system running the gut semi-independently, and all of them communicating with each other and with the brain through intricate feedback loops. And all of this is happening right now, in your body, as you lie there. Your vagus nerve is keeping your heart at a nice, slow resting pace. Your enteric nervous system is quietly processing dinner. Your sympathetic system is idling in the background, ready to spring into action if needed, but content to stay quiet for now. And this brings up something that researchers have only recently begun to appreciate: the idea that the autonomic nervous system isn't just a maintenance crew running basic bodily functions. It's deeply intertwined with emotion, with social behavior, with the way you connect to other people. And the person who put this idea together in the most compelling way is a researcher named Stephen Porges, who in the 1990s proposed something he called the polyvagal theory. And it's one of those ideas that, whether you end up agreeing with every detail or not, fundamentally changes the way you think about what the nervous system is actually for. So here's where Porges started. He'd been studying heart rate variability for years. That's the subtle fluctuation in the time between your heartbeats. Your heart doesn't beat like a metronome. There are tiny variations from beat to beat, and it turns out those variations carry a lot of information about the state of your autonomic nervous system. Specifically, Porges was interested in a particular kind of heart rate variability linked to the vagus nerve, what's called vagal tone. High vagal tone means your vagus nerve is doing a good job of modulating your heart, keeping you calm, flexible, responsive. Low vagal tone is associated with stress, anxiety, difficulty regulating emotions. And Porges noticed something interesting. Vagal tone seemed to correlate not just with physical health, but with social behavior. Babies with higher vagal tone were more socially engaged. Adults with higher vagal tone were better at reading facial expressions, more empathic, more emotionally regulated. And that got him thinking about why the vagus nerve, this ancient, wandering cranial nerve that Lowy had used to slow a frog's heart back in 1921, would have anything to do with how well you connect with other people. His answer, published formally in 1995, was the polyvagal theory, and the core insight goes like this. Porges argued that the vagus nerve isn't really one system, it's two, and those two systems evolved at different times and serve different functions. The older branch, which he called the dorsal vagal complex, is unmyelinated, meaning the nerve fibers lack that fatty insulating sheath that speeds up signal transmission, the same myelin we talked about way back when we were discussing how nerve signals travel. This dorsal branch is ancient. It's found in reptiles, in fish, in almost all vertebrates. And its primary job, when activated strongly, is immobilization, freezing, shutting down. Think of a lizard going completely still when a predator appears, or an opossum playing dead. That's dorsal vagal activation. In extreme cases in mammals, it can cause fainting, dissociation, a kind of metabolic shutdown. The body's last-ditch survival strategy when fighting and fleeing have both failed. The newer branch, which Porges called the ventral vagal complex, is myelinated, faster, and found only in mammals. It evolved much later, and it does something remarkable. It connects the regulation of the heart and the lungs to the muscles of the face and head. Your facial expressions, your ability to vocalize, the tiny muscles in your middle ear that help you tune into human speech frequencies, the muscles that let you make eye contact and tilt your head and smile. All of these are linked neuroanatomically to the ventral vagal system. And Porges's argument is that this linkage isn't accidental. It's the biological foundation of social engagement. When you feel safe, your ventral vagal system is active. Your heart rate is calm and variable. Your face is expressive. Your voice has prosody, that musical, warm quality that signals friendliness. And you're physiologically primed to connect with other people. So Porges proposed a hierarchy. When you feel safe, the ventral vagal system runs the show. You're social, engaged, calm. When you detect danger, the sympathetic nervous system kicks in, fight or flight, just as Cannon described it. And when the danger is overwhelming, when neither fighting nor fleeing is possible, the ancient dorsal vagal system takes over. Freeze, collapse, shut down. 3 levels, layered on top of each other evolutionarily, activated in sequence depending on how safe or threatened you feel. Now, here's the thing you should know. Polyvagal theory is genuinely influential. It's been widely adopted in clinical psychology, in trauma therapy, in understanding conditions like PTSD. Therapists use the framework constantly. The language of ventral vagal states and dorsal vagal shutdown has become part of the clinical vocabulary for understanding how people respond to overwhelming stress. And there's something deeply intuitive about it. Think about your own experience. When you feel truly safe with someone, a close friend, a partner, a family member, notice what happens. Your shoulders drop, your face relaxes, your voice softens, you make easy eye contact. That's ventral vagal activation, at least in Porges's framework. And when you're terrified, really terrified, not the manageable kind of stress, but the kind where you feel trapped and helpless, some people don't fight and don't flee. They go numb. They dissociate. They shut down. That maps onto dorsal vagal collapse. But I should be honest with you. Polyvagal theory is also controversial among neuroscientists. Some of the specific anatomical claims Porges makes have been challenged. The clean division of the vagus into two functionally distinct branches, the evolutionary timeline he proposes, the idea that the dorsal vagus is specifically responsible for immobilization responses. Some researchers argue the evidence doesn't fully support these specifics. A neuroscientist named Paul Grossman has been one of the more vocal critics, arguing that the physiological details are oversimplified and that the theory sometimes gets ahead of the data. And that's a fair concern. Science should be precise about mechanisms, but even critics tend to acknowledge that Porges identified something real and important: the deep connection between autonomic regulation and social behavior. The idea that feeling safe isn't just a cognitive judgment, but a physiological state, something your nervous system determines before your conscious mind even gets involved. Porges actually coined a word for this: neuroception. Not perception, which implies conscious awareness, but neuroception, the nervous system's below-the-radar detection of safety and danger. Your autonomic nervous system is constantly scanning the environment for cues: the tone of someone's voice, the expression on their face, the rhythm of their movements, whether they make eye contact, whether the environment feels predictable or chaotic. And based on these cues, your nervous system shifts states toward social engagement, toward mobilization, or toward shutdown, often before you're consciously aware of why you suddenly feel uneasy or why you feel so comfortable around a particular person. Think about that. You've had the experience of walking into a room and immediately feeling that something is off, even though you can't point to anything specific. Or meeting someone and instantly feeling at ease. That's neuroception at work, at least in this framework. Your vagus nerve and the broader autonomic system are processing social and environmental information and adjusting your physiological state accordingly. And this connects to something we touched on when we talked about predictive processing back in the discussion of Helmholtz and the brain's constant model building. Your nervous system isn't passively waiting for things to happen. It's anticipating. It's running predictions about what's safe and what isn't, based on past experience, and adjusting your body's state preemptively. A person who grew up in an unpredictable or threatening environment may have an autonomic nervous system that's calibrated differently, quicker to shift into sympathetic activation, quicker to detect threat, slower to settle into that ventral vagal state of safety and social engagement. And this is why so much of modern trauma therapy focuses not just on talking about what happened, but on working with the body, with breathing, with the felt sense of safety. Because the autonomic nervous system doesn't respond to logical arguments, you can't reason your way out of a dorsal vagal shutdown. You have to create the conditions, the safe environment, the calm voice, the predictable rhythm that allow the nervous system to shift states on its own. This is also where the vagus nerve has entered popular culture in ways that would probably surprise Walter Cannon or John Langley. You've probably heard of vagal nerve stimulation. It's an actual medical treatment approved by the FDA, where a small device is implanted that sends mild electrical pulses to the vagus nerve. It was originally developed for epilepsy in the late 1990s, and it worked well enough that researchers started wondering what else it could do. Turns out, vagal nerve stimulation also shows promise for treatment-resistant depression, and there's ongoing research into its effects on inflammation, on autoimmune conditions, on PTSD. The vagus nerve, remember, connects to virtually every major organ system. Stimulated in the right way, and you can influence heart rate, digestion, immune response, and mood, all through a single nerve. And then there's the less clinical side. The wellness world has picked up on vagal tone as a concept, and you'll find endless advice about cold showers, deep breathing, humming, and singing as ways to stimulate your vagus nerve and improve your vagal tone. Some of this is grounded in real physiology. Slow, deep breathing with a long exhale really does activate the parasympathetic system through the vagus nerve. That's well established. The mechanism is straightforward. When you exhale slowly, stretch receptors in your lungs send signals through the vagus to your brainstem, which in turn slows your heart rate. It's a direct, physical pathway. You can test it right now if you want. Take a slow breath in, and then let it out even more slowly, and notice what happens. Your heart rate drops slightly. Your body settles. That's the vagus nerve doing its job in real time. But here's what fascinates me about all of this, and what I think ties together so much of what we've been talking about tonight. When you look at the nervous system from the outside, as a diagram in a textbook, it looks like a wiring schematic. Brain at the top, spinal cord running down the middle, nerves branching out to the periphery. It looks like a hierarchy, a command structure, with the brain as the general issuing orders. But the more you learn about how it actually works, the more you realize it's nothing like that. It's a conversation, a constant, multi-layered, bi-directional conversation between your brain and your body, between your gut and your brainstem, between your autonomic states and your social environment. The vagus nerve alone carries more information from the body up to the brain than from the brain down to the body. Roughly 80% of vagal fibers are afferent, meaning they carry signals upward. Your brain is listening to your body far more than it's commanding it. And this brings us to something that's been quietly revolutionizing neuroscience over the past couple of decades. The growing understanding that the brain doesn't work in isolation. That cognition, emotion, memory, perception, all the things we think of as happening in the head, are profoundly shaped by the body they're embedded in. There's a name for this idea, and it's been gaining traction since the early 2000s, though its roots go back further. Researchers call it embodied cognition, and the basic claim is that your mind isn't just your brain. It's your brain, plus your body, plus your environment, all working together as a single integrated system. And one of the most striking examples of this comes from the work of a neuroscientist named Antonio Damasio, who in the 1990s began studying patients with damage to a specific part of the brain, the ventromedial prefrontal cortex, and discovered something that changed everything about how we understand the relationship between emotion and reason. Damasio's patients were fascinating because, on the surface, they seemed fine. They could score normally on IQ tests. Their memory was intact. Their language was fluent. They could reason through abstract logic problems without breaking a sweat. But their lives were falling apart. They couldn't hold jobs. Their relationships crumbled. They made terrible financial decisions, one after another, even when they could clearly articulate what the smarter choice would have been. They knew the right answer. They just couldn't choose it. And the reason, Damasio argued, was that their damage had severed the connection between their rational, planning brain and the emotional signals rising up from their body. These patients couldn't feel the subtle gut-level nudges, the faint tightening in the chest, the whisper of unease, the quiet warmth of a good option, that the rest of us rely on every time we make a decision, usually without even realizing it. Damasio called this the somatic marker hypothesis, and the core idea is beautifully simple. When you've had experiences in the past, good ones, bad ones, painful ones, rewarding ones, your body keeps a kind of ledger, Not in words, not in conscious memory, but in patterns of physical sensation that get tagged to certain situations. So the next time you encounter something similar, before your conscious mind has even finished analyzing the options, your body is already sending up a signal, a faint feeling, a somatic marker. And that feeling biases your decision-making in ways that are usually adaptive, usually helpful, usually pointing you toward the option that's worked out before and away from the one that burned you. Think about that for a second. This means that what we call intuition, that hard-to-explain sense that something is right or wrong, that gut feeling people talk about, isn't mystical. It's not some separate magical faculty. It's your body's accumulated experience being fed back to your brain through exactly the kind of interoceptive pathways we've been talking about. The vagus nerve, the autonomic nervous system, the enteric nervous system with its hundreds of millions of neurons. All of that visceral information flowing upward is part of how you think, not a distraction from thinking, not an interruption of thinking, a fundamental component of it. Damasio's most famous patient was a man he called Elliot, though that wasn't his real name. Elliot had had a tumor removed from his ventromedial prefrontal cortex, and afterward he became a kind of living thought experiment. He was intelligent, articulate, and utterly unable to navigate daily life. Damasio described how Elliot could spend an entire afternoon trying to decide which restaurant to go to for lunch, weighing the pros and cons endlessly, unable to land on a choice. Because without the emotional weighting that somatic markers provide, every option looked equally valid. There was no felt sense pulling him one way or another. Pure reason, it turns out, is not enough. You need the body in the loop. And here's what's really beautiful about this. It connects back to almost everything we've talked about tonight. Remember Helmholtz, back in the 1800s, with his idea of unconscious inference? The brain constantly making predictions about the world based on prior experience? Somatic markers are a version of that, running through the body rather than purely through cortical circuits. Remember the predictive processing framework, where the brain is always generating expectations and checking them against incoming signals? The body's emotional and visceral states are part of that prediction machinery. Your brain isn't just predicting what you'll see or hear next. It's predicting how you'll feel, and when the feeling doesn't match the prediction, that mismatch itself becomes a signal. That's what surprise feels like. That's what unease feels like. That's what the hair standing up on the back of your neck is. A prediction error running through your autonomic nervous system. And remember Porges and neuroception, the unconscious detection of safety and threat? That's the same basic idea, applied specifically to social situations. Your nervous system is reading the environment, comparing it against a lifetime of stored patterns, and generating a felt sense, safe, dangerous, life-threatening, before your conscious mind has any say in the matter. Damasio, Porges, Helmholtz, the predictive processing people, they're all circling the same deep truth from different angles. The body is not a passive vehicle that the brain drives around. The body is thinking. Not in words, not in propositions, but in feelings, tensions, rhythms, and states that profoundly shape every decision you make, every perception you have, every memory you form. This is what embodied cognition really means when you push past the buzzword. And it has practical implications that are still being worked out today. Take something like anxiety disorders. For a long time, the dominant treatment model was purely cognitive. Identify the irrational thought, challenge it with evidence, replace it with a more rational thought. Cognitive behavioral therapy, or CBT, And it works, genuinely works, for a lot of people. But embodied cognition suggests that anxiety isn't just a thought problem, it's a body state problem. The racing heart, the shallow breathing, the tight shoulders, the churning gut. Those aren't just symptoms of anxious thinking. They're part of the anxiety itself. They're feeding back into the brain, reinforcing the threat prediction, making it harder for the cognitive reappraisal to stick, which is why approaches that directly address the body, breathing techniques, movement, yoga, even the vagal nerve stimulation we talked about, can be so effective. They're not just calming the body as a side effect. They're changing the inputs to the prediction machine. They're altering the somatic markers. They're shifting the conversation. Now, let's zoom out even further, because there's one more layer to this story that I think is worth spending some time with, and it has to do with where all of this is heading. We've traced the understanding of the nervous system from Hippocrates and Galen, through Galvani's twitching frog legs, through Kajal's beautiful drawings of individual neurons, through Hodgkin and Huxley's equations, through Lowy's dream about acetylcholine, through Hebb's learning rule and the discovery of long-term potentiation, through Hubel and Wiesel's orientation columns and Penfield's homunculus, through the autonomic nervous system and the vagus nerve and the gut-brain axis. That's roughly 2,500 years of slowly building understanding. And the thing that strikes me most about the whole arc is how the story keeps getting more complicated in a very specific way. It keeps getting more connected. Every time scientists thought they'd found the fundamental unit, the nerve, the neuron, the synapse, the circuit, they discovered that the unit only made sense in the context of a larger system. Neurons don't work alone. They work in networks. Networks don't work in isolation. They're shaped by neuromodulators bathing whole regions of the brain. The brain doesn't work independently of the body. It's in constant dialogue with the gut, the heart, the immune system. And the body doesn't work independently of the environment. It's reading social cues, responding to temperature and light and sound, being shaped by experience from the moment of birth. This is one of the big frontiers right now. Understanding the nervous system not as a collection of parts, but as a dynamic, adaptive, self-organizing system that extends beyond the skull. People are studying the microbiome gut-brain axis, tracing how the bacteria in your intestines can influence your mood and cognition through vagal signaling and immune pathways and the production of neurotransmitters. Remember that 95% of your body's serotonin is in the gut. Researchers are now finding that specific strains of gut bacteria can alter serotonin production, and that this actually changes behavior in animal models, anxiety levels, social behavior, even memory performance. The implications for understanding depression, autism, and neurodegenerative diseases are enormous, and we're really just at the beginning of mapping those connections. There's also the field of neuroplasticity, which has exploded in the last few decades. We touched on this when we talked about Hebb and long-term potentiation, the idea that synapses strengthen with use and weaken with disuse. But the modern understanding goes much further. The brain doesn't just tweak the strength of existing connections. It grows entirely new ones. It prunes old ones. In some regions, it even generates new neurons throughout life, adult neurogenesis, something that was considered impossible until the late 1990s, when researchers like Elizabeth Gould and Fred Gage demonstrated it in the hippocampus, the same structure that was so devastated in patient HM. The brain you have right now, tonight, is not the same brain you had a year ago. It's been physically remodeled by everything you've experienced, everything you've learned, every habit you've practiced, every emotion you've felt. The hardware changes in response to the software. which is a very strange thing if you think about it from an engineering perspective. Imagine a computer that physically rewired its circuits based on the programs you ran on it. That's what your brain does every single day. And then there's the question of consciousness itself, which is maybe the deepest question of all, and one that neuroscience still can't fully answer. We understand a tremendous amount about how neurons fire, how signals propagate, how networks process information, how the brain predicts and corrects and learns. But we still don't have a satisfying explanation for why any of this is accompanied by subjective experience. Why does it feel like something to see the color red? Why does pain hurt, rather than just being an information signal that triggers avoidance behavior? This is what the philosopher David Chalmers called the hard problem of consciousness in 1995, and it remains genuinely hard. There are theories: integrated information theory, global workspace theory, higher order theories, and they're all interesting, and none of them has achieved consensus. It's humbling, honestly. After all these centuries of progress, after Galvani and Kajal and Hodgkin and Huxley and all the rest, the most fundamental question about the nervous system, how does a 3-pound mass of cells generate the experience of being you, remains open. But here's the thing. I don't think that's a reason for frustration. I think it's a reason for wonder. Because look at how far we've come. A few hundred years ago, people thought nerves were hollow tubes carrying animal spirits. Now we can describe the exact sequence of ion channel openings that produce an action potential. We can trace the molecular cascade that converts a fleeting experience into a permanent memory. We can explain why your pupils dilate when you're startled, and why a deep exhale slows your heart rate, and why damage to a specific patch of cortex can make you unable to recognize faces while leaving the rest of your vision perfectly intact. That's extraordinary. That's a species figuring out, piece by painstaking piece, how its own most complex organ works, using that very organ to do the figuring. There's something almost paradoxically beautiful about that, and the practical payoff is real. Every drug that treats depression or epilepsy or Parkinson's disease, every deep brain stimulator, every cochlear implant that lets a deaf child hear for the first time, every prosthetic limb controlled by neural signals, all of it flows from the centuries of slow, patient, often frustrating work we've been talking about tonight. From Swammerdam carefully dissecting a frog in a Dutch laboratory in the 1660s, to a modern neurosurgeon threading an electrode into a patient's subthalamic nucleus to quiet the tremors of Parkinson's. It's one long, continuous thread of curiosity and persistence. So as you're lying there, maybe drifting a little, maybe already half asleep, I want you to think about this. Right now, your nervous system is doing all of the things we've talked about, all at once. Your brainstem is regulating your breathing without you having to think about it, adjusting the rhythm automatically as you relax. Your vagus nerve is carrying signals from your gut and your heart up to your brain, reporting that things are calm, that you're safe, that it's okay to let go. Your parasympathetic system is gently ascendant, slowing your heart rate, lowering your blood pressure, shifting your body toward rest and repair. Your cortex is processing these words, but more and more loosely, letting them wash over you rather than gripping each one tightly. Somewhere in your hippocampus, the day's experiences are beginning to be consolidated, synapses strengthening here, weakening there, your brain quietly editing and filing and reorganizing while you sleep. Your sensory neurons are still reporting the weight of the blanket, the temperature of the air, the faint sounds of the room. But your thalamus is gradually closing the gate, filtering out more and more of that input, letting you sink deeper. All of this is happening without any effort from you. Billions of neurons, trillions of synapses, an entire orchestra of chemical and electrical signals, all playing together in a coordination so intricate that no engineer has ever come close to replicating it. And the most remarkable part is that this system built itself. From a single fertilized cell, following instructions encoded in DNA, your nervous system wired itself into existence. Neurons migrating to their proper positions, axons growing toward their targets, synapses forming and pruning and strengthening based on experience. No one designed it from the outside. It emerged, and it's still emerging, still changing, still adapting, right now, tonight, as you rest. So let that be the last thought you carry with you. You are, in this very moment, the most sophisticated nervous system that evolution has ever produced, doing exactly what it was built to do. Winding down, letting go, drifting into the restorative quiet of sleep. The conversation between your brain and your body is shifting to a gentler register now, the signals softening, the rhythms slowing, everything settling into that ancient, familiar pattern that every human who has ever lived has known. You don't have to understand it all. You don't have to hold on to any of it. It's all still there, woven into the tissue of you, working quietly in the background, taking care of things while you rest. So just breathe. Let your body do what it knows how to do, and sleep.